Morphology. Morphology of E. coli bacterium. Now, in the morphology, we will discuss about first their normal, you can say general morphology. So first heading you should make the general morphology. Morphology means their appearance, okay? So generally, these bacteria, they are the rod-shaped bacteria or bacillus bacteria, yeah? So if you talked about their shape, they are the bacillus bacteria, a rod-shaped bacillus bacteria, okay? And they are gram-negative. Yeah, gram-negative means in their cell membrane, okay, their cell membrane is thinner as compared to the others, and their cell membrane is made up of the lipopolysaccharide. Sorry, cell wall is made up of the lipopolysaccharide. So lipopolysaccharide layer is there here, lipopolysaccharide. Polysaccharide. Okay, and they possess both pili and embryo. Pili and embryo, and their pili part are the virulence part. Okay, they producing the antigen, and that antigens are, you can say, dangerous. That antigens is the dangerous. Fine, no problem. Now let's draw the diagram of E. coli and understand how they appear. Okay, how they look like. So as I said that they are present in the human's colon. They are present in the human colon. So suppose this is your GIT tract. Yeah, this is your GIT tract. Here is your stomach. These are intestine. Okay, and here is the large intestine part. So these bacteria normally, if we talk about the normal flora, then they are present in the colon. Yeah, they are present in the colon. Fine, but when you acquired the pathogenic strain of E. coli, which is strain? Pathogenic strain. Write it down. Pathogenic strains means those type of E. coli which have the virulence, which have the disease-causing capacity. Fine. So if that strain you acquired either from the oral route, either from the oral route or from the fecal route. So oral fecal route are the, you can say, pathway by which this bacterium enters in our body. Fine, no problem. Now these bacterium, pathogenic bacterium, they enters. Now we are enlarging their shape and studying their morphology, their general morphology. Since it is a gram-negative rod, okay, fine. They are generally, most of the, you can say, E. coli are capsulated. Write it down. But certain are also non-capsulated. So most E. coli are capsulated. Okay, most E. coli are capsulated, and there are certain types where the capsule is absent. The capsule is absent. Fine. Now, as I said, as this bacterium, they have the plasmid, okay, and when these plasmids they have, that plasmids is the one part of bacteria, okay, which is like it's a double standard circular DNA, okay, which is independent from their parental DNA, okay, parental genetic material. And whenever these you can say the plasmids, okay, whenever these plasmids, they acquire certain, you can say, a gene, okay, they synthesize certain proteins which make the bacterium pathogenic, okay, which make that's the one way to make the E. coli pathogenic. The another way is to, if you know that bacteria also suffer. Not bacteria always causes a disease in the humans. They itself suffer. They still suffer from the what? A bacterial virus. And the name of the bacterial virus is, it's a bacteriophage. So whenever the E. coli is affected, whenever the E. coli is affected by the bacteriophage, okay, by the bacteriophage, E. coli become infectious. Whenever bacteriophage is affect the E. coli bacterium, okay, these bacterium become the pathogenic. So what are the reason behind this E. coli to being a virulence or generating a virulence factor or being a you can say disease causative agent? It's a two factor. The one is either changes in their plasmid genetic material, or either the E. coli is affected by the bacteriophage or bacterial eating, you can say, virus, bacterial affecting virus called as the bacteriophage. There are various type of bacteriophage, okay. And now the another thing is that apart from having all these things, bacteria have the pili, it's have the pili, and it's have the fimbriae, it's have the fimbriae, and these, you can say, the pili or fimbriae, they possesses certain capsules, okay. Over these capsules, there is a presence of certain antigen. We should write it down, the name of the antigens called as the K antigen, okay, called as the K antigen and also known as the capsular antigen. Now, the flagella contains the H antigen, H antigen, fine. And there is also a liposaccharide part of the cell wall. They are containing the O antigen. Which antigen? O antigens. So there are three types of antigen. One is called as the K antigen. This K antigens is also known as the capsular antigen. Got the points? No problem. Another one is called as the flagellar antigen, or this flagellar antigen is called as the H antigen. Fine. And there is the lipopolysaccharide. Okay, lipopolysaccharide are also called as the endotoxin. So basically, this is the not the antigen. This lipopolysaccharide, okay, it's a endotoxin. It's a endotoxin. As I said initially, that this E. coli produces the two types of virulence factor. The one is to synthesize the antigen, and another one is to synthesize the endospore. Fine, no problem. So this is about their general morphology. Once again, what we studied that in the morphology, first we discussed the general morphology. In the general morphology, we discussed they are the facultative anaerobes, okay, and if they talked about their shape, they are a bacillus shape. Okay, they are the bacillus shape. They are the gram-negative bacteria. They have the lipopolysaccharide, a thinner cell wall. There is the presence of fimbriae and the, you can say, pili and the fimbriae. And remember, most of the E. coli, if we talk about their motility, they are the motile. They are the motile bacteria. Fine. And most E. coli are the capsulated, some of the non-capsulated. Now, if we talked about their pathogenic strain, we acquired this pathogenic strain, okay, by the fecal-oral route or oral fecal route. Fine. Generally, normally, these bacteria are located in the colonic lesion, in the colon. Fine. But we acquire the pathogenic strain from either oral route or you can say the fecal route. Got the point? Now, then we seen there, you can say, structure. Their anatomy lies with the presence of different types of antigens called as the K antigen, also called as the capsular antigen. Then there is the H antigen. Then there is the endotoxin. Okay, and this E. coli become pathogenic. Okay, just because of the two reasons. The one reason is, very good. It is affected by the bacteriophage. Another reason could be, another reason is their plasmids make certain changes which result into synthesis of certain endospore, which is a pathogenic type. Got the points? No problem. Now, after discussing the general morphology, we also discussed hand by hand. We also discussed about their antigenic morphology. No problem. Now, let's discuss about their colony morphology. Their colony morphology. So their colony morphology is easy to understand. Why? Just because they are the lactic acid producing bacteria, it means we can culture this bacteria on the which agar plate? Yeah? I'm going to impress by your answer. Yeah? It's a very good. It's a McConkey agar. McConkey agar. So when you taking the specimen or sample and you grow on the McConkey agar, it shows which color? Yeah? Pink color. Very good. It shows the pink color. Fine? No problem? Now, after having said all these things about their morphological characteristics of E. coli, now let's move ahead and discuss their strain.